Now I am going to make a glass needles with a very fine tips to move around the nerves without damaging it, and also we can use this glass needles to separate the PD organ from the main nerve. Then you can use this and also like hold the tip, but don't push it very hardly because it will break the glass. Then after that, but be careful, like if you hold the glass too much on the fire, it warms it up. Then pull it like this. Now I'll run this over the fire to make a smooth ending here. Now we got this. It should have a nice smooth end. Then remember to pin it like you have a way to move the dactylopodite all the way here. Then now we have a good space. And sometimes it's very long, then you can trim the tip of the dactylopodite. Then we should like pin it here, and also on this part too. Then you can see that, let's remove the small pin. Then you can see that you can move the dactylopodite all the way. Then now we should know that the opener muscle is located at the dorsal region, and the closer muscle is located at the ventral region, and the PD organ crosses from dactylopodite to propodite, and it attached to the tendon of the closer muscle. Therefore we can remove the opener muscle here, but we have to be very careful about the closer muscle here. Then now I'm going to remove the opener muscle to get more view in order to see the nerves. Then I'll grab the opener muscle with the forceps, but be sure that the nerve is not, you shouldn't like pull the nerve because it damages everything. Then you can cut the muscle here and the tendon also. Then you can go all the way with your scissors then and cut it here like this. Then you see that all muscle fiber is detached, and pull it all the way because we don't need this part, you see. Also we can cut it off here. Now I'm going to use small pins in order to separate the nerve bundle from the closer muscle, but you should be very careful not to pin the nerves because it will damage it. Then here we can see the nerves here, bundle of nerves, but like I said, you should be very careful here. Then now I can pin it, and you can see here a nice. Then you can use your glass rod to separate the small nerve from the bundle nerve. Then we can push the nerve, like the nerve bundle with the pin, and put the pin here. Then you can see a small branch of nerves goes to the PD organ here. Then now we are done with the dissection of this prep. Then let's record the electrical activity. Then I'll put the dish on this dissecting scope. Then we have to wax it. Be sure that it's fixed like this. Then this is the ground wire here, you see, and we should plug into the middle one here. And also we have two wires here. Then those two wires goes to the suction electrode. You see that there's two wires, one outside and one inside here, and we should plug in these wires into the other holes, but make sure that there is no contact between these two wires. Okay, then also you can see the wire, this wire inside the suction electrode. Then the saline should be up until it reaches the inside wire. Then now we should put a negative pressure on the syringe here. Then you see that the saline goes up here. Now I put the ground wire inside the solution. It should be away from the prep, because when it moves, in order not to cause a problem for you, then now it's inside the saline. Then you see that this wire from the head stage is going here to the differential amplifier. Then let's turn on differential amplifier here. Then follow the lab manual for settings. Then you see that this wire, this is the output wire, it goes to the analog digital board or AD board here. Then this board is connected with the computer here. Then now let's open the lab chart that we needed to acquire our data here. When the window opens, then go to setup here, then channel settings, click on this one. Then you see that you have 8 channels, but we don't need 8 channels, we just need 1 channel. Then just type 1 here and enter. Then you see that you have just 1 channel. And also go click here and choose the 200 millivolt gain, and the acquisition will be about 10K. And also click on this, the channel 1, and go to input amplifier. Click on this one. You see that the window input amplifier, and here it's already checked. Then you can check it on, then click OK. Then now let's suck the nerve into the suction electrode to see if you get electrical activity here. Then click on start. Then now the nerve is in the suction electrode. Then now we want to record the electrical activity. Then this is the dactylopoid. If you move the dactylopoid all the way 180 degrees to see what happens, and also if we open it slowly, then we see the electrical activity also. And also if we just open it 45 degrees to see what happens. Then now I'll move the dactylopoid all the way 180 degrees slowly and hold it. Then go back again, close it. You see there's no activity. And also this time 45 degrees, and you see, then hold it, then close it again. And this time I will move it very fast, 180 degrees, hold it, then again close it. Then we got the data. Now we have to analyze the data. Then we can count the frequency of spikes for like specific time, like 1 second or 2 seconds. Then we can compare the like when you open it 180 degree with 45 degree to see the difference in frequency of the spikes. Then if you look at the amplitude of the spikes carefully, you see that there are different spikes in terms of their amplitude, then I will let this on you to figure out why you get some kind type of like big amplitude spikes and small amplitude spikes. Then now we are done with the electrical activity recording, and then after that we wanted to stain the PD organs with the methylene blue. Then sometimes we have to remove some of the muscles to see the PD organ. Then after, when we are done with the further dissection by removing more muscles here, then we don't need the saline. We have to throw this away. Then we have methylene blue here. Then we can pour the methylene blue into the petri dish. Then after that, you can leave it like this for 10 minutes. But if you wanted to get better staining, then you can leave it overnight. Then after that, you can look at it to see the PD organ, all these cell bodies of small and also large cell sensory cell bodies. Then leave it like this. You can do. Then after that, when you are done with the staining, then you can pour the saline back into the this small beaker to use it again, and also you can use a fresh saline to wash it again. Then if you wanted to get like the good staining, then you have, like I mentioned before, you have to leave it overnight. But it's better to put it inside the refrigerator, because when the methylene blue is cold, then you can get better staining. But when it's warm, then it will like destaining. Then therefore it's better to use a cold saline, like cold methylene blue, or put it inside the refrigerator overnight.